Okay, hello everybody, and this is finishing up the lecture on molecular biology. We had talked about transcription, and now I wanted to finish up with mRNA processing in eukaryotes. So bacteria don't process their mRNAs. Transcription and translation occur at the same time and in the same space, and so there's really no time or place for them to modify RNAs. However, eukarya add a 5' cap. They splice out introns and polyadenylate the 3' tail. So we talked a little bit about this. As you'll remember, there's a 7-methylguanosine 3' cap at the 5' end. It's an unusual looking nucleotide in that it has a triphosphorylated nucleotide that is added to the 5' carbon of the first nucleotide that was incorporated during transcription. So the 5' cap is going to protect the RNA from degradation. It's also going to promote the translation. In addition to the 5' cap, you can see the untranslated regions in the 5' and 3'. This transcript at the top here is not showing any introns. We'll talk about those, and there's a long poly A tail that's not encoded in the DNA, but it's added after transcription by poly A polymerase. And this also helps protect the RNA from degradation and promote translation. So that's where we stopped. This is a slide on splicing, and this was discovered, splicing was discovered in the 1970s. Primarily out of the lab of Phil Sharp. They were doing some hybridization studies, incubating DNA and RNA together and looking to see what those hybrids looked like. And the figure shown in A is a micrograph, an electron micrograph of the hybrid. And what they noticed was that there were segments of DNA that were not present in the fully mature RNA. So that's shown schematically in section B over here, where the DNA is in red and the RNA is in yellow. And those segments of DNA that loop out and form the lariats, those are introns that were transcribed in the precursor RNA, but they've been spliced out. And so you can see here in this long species of DNA that quite a bit of it ends up spliced out. And so the takeaway message is that genes and RNAs can often differ in length, and so that these introns after transcription are removed. And so the ultimate size of a mature RNA is smaller than the DNA in many cases where splicing occurs. The machinery that carries out the splicing is known as the spliceosome. It's small nuclear RNAs and proteins, much like the ribosome. It's composed of numerous RNAs and many proteins. It's a large macromolecular machine. So this is often called SNRPs, so small nuclear ribonucleoproteins, or the spliceosome. So you can see here in the figure below that DNA that encodes a gene has a promoter region which drives transcription, and then in this case there's 3 exons and 2 introns, and then 2 untranslated regions, the 5' and the 3' untranslated region. So looking at this, we'd be looking at the template strand. Strand is transcribed by RNA polymerase II to give the 3 exons and 2 introns, and the spliceosome works on that RNA transcript to splice it and stitch those 3 exons together and remove the 2 introns. And so the mechanism of splicing is shown here. There are specific nucleotide sequence at the boundaries of the introns and exons. A G and a U especially shown here, and that's bound by one component of the spliceosome. And so once that site is recognized along with an A, which is in the intron, by another portion of the spliceosome that brings the whole complex together so that these SNRPs assemble. And once they're fully assembled, that's when it gets the name the spliceosome. The loop is formed and the intron is cut at both borders. And so in step 4 here, you can see that the intron is released as a lariat and the exons are joined together, and you have a spliced mRNA. And splicing occurs within the nucleus prior to export, prior to translation. So if we're to compare transcription in bacteria and eukaryotes as a general summary, what you can see here, there are key points to note in that bacteria have only one RNA polymerase. Eukarya have 3, RNA polymerase 1, 2, and 3. And although these have generally the same features as bacteria, and that the overall architecture is kind of the same, it looks like somewhat of a Pac-Man structure. Eukarya polymerases are specialized for their individual functions. So we talked about RNA polymerase 1 transcribes most of the ribosomal RNAs, and then RNA polymerase 2 transcribes the mRNAs and the microRNAs, and RNA polymerase 3 transcribes the short RNAs like the small ribosomal RNA and the tRNAs. Plants have RNA polymerase 4 and 5, but we're not going to talk about those. Those are very specialized, and they deal with really unique features of plant genomes. So we'll leave that for another class. Their promoter structure in bacteria typically contains a -35 box and -10 box. It looks like a TATA region. And these are contained in front of most genes in bacteria. Eukarya have more complex promoter structure, and it's variable, so it often includes a TATA box, which is about 30 nucleotides from the start of the gene. But then there are also a lot of other promoter elements that control transcription, both in a tissue-specific manner, in a timing-specific manner, and also in response to a lot of different stresses. So the extent of transcriptional control is much more complex in eukarya. And then bacteria, we talked about sigma factor as one of the primary factors responsible for recruiting RNA polymerase. So it recognizes those 2 elements in the promoter and will recruit RNA polymerase. So there's different versions of sigma, but they all serve the same function in bacteria. And then in eukarya, there are many basal transcription factors, all of those TF2, A, B, C, D, that bind sequentially to the DNA and have a role in recruiting RNA polymerase and orienting RNA polymerase properly, and then allowing transcriptional firing to occur at the proper time, and allowing RNA polymerase to release into the elongation phase of transcription. And then finally, we just covered it, but RNA processing. So there's really no RNA processing in bacteria. The translation co-occurs with transcription. And then in eukarya, there is the enzyme catalyzed addition of the 5' cap. There's splicing through the spliceosome, and enzyme catalyzed addition of the poly A tail. And so with that, it's kind of where I want to leave you with the molecular biology section. We will pick up with translation in the next lecture.